This tutorial is on the Calvin cycle, which is the second stage of photosynthesis. So if you take a look down in the right-hand corner, I've written the equation for photosynthesis to just kind of review what we've gotten to so far. So you'll see it's water plus carbon dioxide in the presence of light in an autotroph will create C6H12O6, which is glucose, and oxygen. So far in the light reaction, we have taken water in through the roots of the plant, and we've split the water to grab the electrons off of it to go through the electron transport chain, first going into the photosystem 2 and then the electron transport chain in the light reaction. We've also produced in the light reaction oxygen from that same splitting of the water molecule. But as you see, we have yet to make the major product of photosynthesis, which is the glucose. So let's take a look at how that happens. This Calvin cycle, just a reminder, happens in the stroma. That's that fluid that's around the thylakoids. So as you remember from the last chapter, in the Krebs cycle, the definition of a cycle is that the products of this cycle are often reused in the next go-round of a cycle. And that's actually what's happening here as well. So let's take a look. We begin with carbon dioxides, which came in through the stomata. Remember, the stomata are the little openings in the bottom of the leaf in which carbon dioxide can come in and flow into the stroma. So what we're going to do is when this carbon dioxide comes in, we're going to attach it to the previously made molecule, which is called RUBP. There's actually 3 of them. RUBP stands for ribulose 1,5-phosphate. Biphosphate. You don't need to know that, but just that's what RUBP stands for. So if you look at RUBP, you notice that there's 1, 2, 3, 4, 5 carbons in each RUBP. Well, what each RUBP is going to take is one of those carbons from the carbon dioxide. So we end up with, when those two combine, 1, 2, 3, 4, 5, 6 carbon molecules, 3 of them. However, those 6 carbon molecules are not very stable at all. And so what happens is they split. And when they split, what you end up getting is these 3 carbon molecules, 1, 2, 3, which are called PGA. PGA stands for phosphoglyceric acid. It's only an intermediary form, but it's important to the process of the Calvin cycle. But you'll notice that there are 6 of these. Okay, so we still have the original 18. There were 18 carbons here. It's kind of important on the Calvin cycle to keep track of your carbons. We still have 18 carbons, but instead of 3 chains of 6, we now have 6 chains of 3. So we've got these 6, 3-phosphoglyceric acid as the intermediary step. And what we'll do is we'll be taking 6 ATP. You may be wondering, hey, where did those ATP come from? Well, these ATP came from as products of the light reaction. We're going to take those 6 ATPs and take the energy from that. And as you know, in the process of taking energy from ATP, we're going to break off that final phosphate and change it into ADP plus P. We're also going to be taking the energy and electrons from NADPH. Again, if you remember from the last tutorial, these were made in the light reaction. And when we take the electrons and the hydrogens from there, or the protons, I should say, we end up with 6 of the NADP+. Again, remember, this is all about cycling, so we're not going to lose the ADP plus P or the NADP plus. That's actually going to be going back to the light reaction, which I'll talk about in a minute. So after we've done this, this molecule, G3P, which is glyceraldehyde 3-phosphate, it may look like the 3-PGA that we talked about earlier, but you'll notice the yellow highlighting. That's to show you that there's energy. And remember, we just got the energy from the ATP and the NADPH. So then what occurs is we take one of these glycerol 3-phosphates for each turn of the Calvin cycle. Once we get 2 of those, so this is a 3-carbon molecule, once we get 2 turns of the Calvin cycle, this is what's going to go on to make our product of C6H12O6. However, as you know, a cycle has to continue, and we didn't end up with, when we take the 1 G3P off and end up with 5 G3Ps, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 carbons at this point. Okay? And so now what we have is we need to rearrange those carbon molecules to get to those RUBP. So when the next carbon dioxides come in, we can use those to go through the cycle again. But in order to rearrange these 5 molecules of 3 carbons, we're going to need some more ATP. Again, we got this from the light reaction. And we're going to use that to create 3 5-carbon molecules, again still 15 carbons, 3 molecules of RUBP. And then it starts all over again with a new 3 molecules of carbon dioxide to do this again. And as I said before, once we've gone through the Calvin cycle twice, we are able to use those 2 G3Ps that came out of those 2 cycles to create our end product, which is glucose, C6H12O6. So as you can see, the Calvin cycle doesn't directly produce glucose. It's actually producing these G3Ps. But then what happens is the cell is able to take the G3Ps and combine them to make the glucose. So let's take a look. This finishes the Calvin cycle, but I want to take a look at the overview of photosynthesis to kind of show you how the light reaction and Calvin cycle depend on one another. So let's take a look at a second slide. This slide they call the photosynthesis roadmap, and I think it's a really excellent slide showing you the relationship between all of the parts of photosynthesis. So let's just take a look real quick. In the light reaction here, you notice it's in these thylakoid stacks, so that's reminding you that that's where it occurs. More specifically, it's occurring in the membrane of the thylakoids. So we've got the light that comes in, and you need that to start all of the light reactions. That's an input. You also have the water come in as an input. Remember, we're splitting that up at the very beginning of the light reaction to give electrons to photosystem 2, and we leave some hydrogen ions or protons in the inner part of the thylakoid. And then also in that splitting, we're also producing oxygen. And that's one of our waste products, and this is why plants and autotrophs are so important to us, because they're producing oxygen for us to breathe. But you'll also notice in the light reaction, we're producing ATP and we're producing NADPH, an electron carrier. So the great thing about this, or the whole cyclical nature of this, is that we're taking the ATP and we're using it in the Calvin cycle, and we're taking the NADPH and we're using it in the Calvin cycle. We also, if you remember, have carbon dioxide coming in through the stomata. Remember, don't confuse stomata with stroma, because it is coming into the stroma. That's what this stuff was. And it goes through the Calvin cycle. And the whole point of the Calvin cycle is to take the carbon dioxide and these ATP and NADPH that came from the light reaction and produce, remember we said that there were 2 G3Ps and we end up producing sugar from it, that glucose. In the process of going through the cycle, we're producing ADP plus P, and we're also producing NADP plus because we're breaking up the ATP and we're taking the electrons and protons off of NADP plus, and the cycle will continue to go and go. But what you'll notice is the product of glucose is considered waste, because the plant doesn't need it anymore, or will need it in cellular respiration but doesn't need it for photosynthesis. But the ADP plus P that is produced by the Calvin cycle is going to be used in the light reaction, and then NADP plus that was used in the Calvin cycle, or produced, goes into the light reaction. So as you can see, this is all really cyclical. And the understanding is you can have the Calvin cycle occur without light, but only for so many times, because you need the ATP and NADPH from the light reaction to make it occur. So there may be some ATP and NADPH floating around in the stroma, which would allow you to have the Calvin cycle occur a few times, but eventually it runs out of that, and then it can't continue. And so this is why photosynthesis can't continue to make sugar in the dark. So that's photosynthesis. I hope these tutorials have helped you understand these things a little bit better.